So moving on to objective 2, we're going to compare and contrast the histological organization of the nephron and the functional aspects of each segment of the nephron. So I want you to think about this as the mainstay of our talk about the renal system, is you really want to think about the nephron and think about each unit, each part of the nephron. And later on, we're going to add the functional aspects. So it's really important for you to understand the anatomy, the location of these structures and how they're connected to one another, so that when we add that layer of the functional aspects, you're not lost in terms of how that function applies to that structure. Okay, so again, that form follows function. Each part of the nephron is shaped differently, has different size of cells, different types of cells, different adjacent structures in order to complement or to adapt the specific function of each of those parts of the nephron. So again, that form follows function that we see for other areas of the body as well. So the structural and functional unit of the kidneys is called the nephron. This is where the waste, and when we say waste, this simply means what has been filtered out from the blood. So kind of the backstory or the context for the renal system is basically the production of urine. And so urine is basically taking our blood, taking our plasma, filtering that, physically squeezing it through the sieve-like structure called the glomerulus, and then removing the watery component, leaving behind the cells and the proteins, and removing that waste that is in the watery component. And that is what we mean by waste, right? When we filter out our blood, take away any toxins, extra fluid, extra ions that are not needed, and remove that, and then keep our cells and our erythrocytes behind. So we're going to remove that waste. We're going to filter that waste out here at the glomerulus. And so we want to understand the structure of the glomerulus and how that filtration can be done. So we've got the glomerular capsule, and the glomerular capsule constitutes the glomerulus itself along with the Bowman's capsule. Okay, so there's the two structures that form the renal corpuscle, right? You can see that here. The renal corpuscle formed by the glomerular capsule and the glomerulus itself, and that is basically going to be responsible for filtering out our blood. That will then get communicated to the proximal convoluted tubule, so that's the initial part of the tubular structures. We then go to the nephron loop, so this downward projection here is the nephron loop, also called the loop of Henle. We then communicate to the distal convoluted tubule. And we really want to understand the relationship between the proximal convoluted tubule, the distal convoluted tubule, and the renal corpuscle, which is really the glomerulus and the Bowman's capsule. Now, the Bowman's capsule is also called the glomerular capsule, okay? So basically, the distal and proximal convoluted tubules are not separate structures the way we will draw them out for simplicity's sake. They both are coming in contact to each other, and they both are coming in contact to the renal corpuscle, where we have the glomerulus and the glomerular capsule. All right. So let's draw some of those structures out here. Again, I'm going to draw them out in a simplified manner, but we still want to understand how they are all coming into contact with each other as we add on the functional aspects. So let's start by looking at an overview of the nephron. Okay, and so we can first start by looking at some of the vessels, the capillaries that feed into the glomerulus, okay? And we'll talk about the major branches of the other parts of the vasculature, but we first want to identify the afferent arteriole, and we'll talk about the vessels that lead up to the afferent arteriole, as well as the efferent arteriole. And if you remember from previous knowledge, whenever we see afferent starting with an A, this is a vessel or a structure, if we think about the neurons, that is going towards something, whereas the efferent branch is going away. So the direction of flow of the afferent is toward the glomerulus. The direction of flow of the efferent arteriole is away from the glomerulus, okay? Then we want to talk about the glomerulus itself. And the glomerulus is basically a ball of capillaries, okay? And I'm going to draw it like this for simplicity's sake, but imagine you took a capillary and you crumpled it up upon itself and you shoved it down into another structure. That is what the glomerulus is. It's a ball or a tuft of capillaries. Okay. We then want to talk about the glomerular capsule, okay? And so we're shoving this ball of capillaries down into this glomerular capsule. And so the capsule is going to have two walls. The wall that comes immediately into contact with our ball of capillaries or our glomerulus, and then the parietal layer of that wall that comes into contact with the space that is being provided in between those two layers. Okay, so I'll say that again. This is our glomerular capsule here. Another very common term for this is the Bowman's capsule. Okay, and it is really formed by two layers. So very similar to the peritoneal cavity or the pleural cavity. It's got the parietal layer. So this is the outer parietal layer. Okay, this is the inner visceral layer here. So this is our visceral layer. And then this is going to be our peritubular space. Okay, so because of the two layers of this wall or two layers of this capsule, we now have a space inside that is our peritubular space. Okay, sometimes called just the tubular space. All right, so this is kind of the proximal part of our nephron, okay? And all of this together is called the renal corpuscle. So the glomerulus plus the glomerular capsule is called the renal corpuscle. And this is where the filtration happens. So this is the first part of the structural or the structures of the nephron that are responsible for filtering our blood across this membrane. And then we begin with the filtrate, which is the initial substance that will then become our urine, okay? Now, that's not the end of urine production. We're going to take that filtrate, which is in this space, and we're then going to send it through the rest of the nephron, which is where we have the tubular structures, okay? So we have the renal corpuscle, which is the initial part of the nephron, and then the rest of the nephron is where we have the tubular structures. And this is really where we fine-tune that filtrate. So we've first filtered our plasma across this membrane, and we're going to then send it through a series of secretion and absorption and fine-tuning that filtrate, which will then become our urine. So the first part of these tubular structures is the proximal convoluted tubule. And convoluted just indicating its nature, right? The fact that it's kind of wounded or it has these bends and twists and turns. So here is our proximal convoluted tubule. And then it goes into this dip that is called the loop of Henle. This has a lot of other terms, by the way. You may, if you're reading this up in other texts, you may see this referred to as several different terms, nephron loop, other terms, but we're going to use the loop of Henle. right? So this is the loop of Henle, or the Nephron loop. Okay. We then look at the distal convoluted tubule. So just like we had a proximal convoluted tubule, we then have a distal convoluted tubule. Okay. And before we go any further, I want to explain the anatomy of these two structures. So I kind of alluded to this earlier, but we're drawing it out in this way for simplicity's sake, but we want to understand that the proximal convoluted tubule and the distal convoluted tubule actually come into very close contact with the glomerulus, okay? Really important relationship that we're going to expound on later on. So while we're drawing it out in this sort of, sequential flow, for simplicity's sake, this is all real bunched upon each other, right? These structures are kind of bunched together such that the proximal tubule and the distal tubule come into contact or are folded down where they can be into close contact with the glomerulus. Alrighty. We then have the collecting duct. So from the distal convoluted tubule, we get into the collecting duct. Oops. This is the major duct where we really start to communicate that urine from the nephron and the tubular structures to the renal papilla, and then to the renal, the minor and then major calyces toward the renal pelvis. Okay. So this is the collecting duct. And then we move towards the minor calyx. So after we have fine-tuned that filtrate, we now have urine. It's going to be collected into the collecting duct, where it will then communicate down towards the minor calyx toward the ureters. All right, so this is the direction of flow of our urine, or of our filtrate that is becoming urine. Okay. The last thing I want to point out here before we move along is the difference between where these structures are located. So we have this separation here, so this imaginary separation where we have the renal corpuscle involving the glomerulus, the Bowman's capsule, the proximal tubule, the distal tubule. All of these structures are located up in the cortex of the kidney, all right? So this is located in the renal cortex, that lighter or paler staining region towards the periphery of the kidney, whereas the loop of Henle dips all the way down into the renal medulla, okay? As well as the minor and then major calyx. So this is down in the renal medulla. Alrighty. And so these, the length of these loops of Henle are really what form the renal pyramid. So we're going to talk about the renal pyramids going forward, those triangular shaped structures at the central part of the medulla of the kidney, and that is being formed by the rays that are being projected from the loops of Henle. So the loops of Henle are forming the renal pyramids. So just to connect some of these microscopic nephron structures to some of the gross things that we can see on the kidney just by looking at it.